Hi, my name is Shannon Oda, and I'm an assistant professor at Seattle Children's in the University of Washington. And today I will be talking about overcoming cell therapy barriers in the suppressive tumor microenvironment with engineered proteins. Here are my disclosures. So it's an exciting time to be in the cancer immunotherapy field. There's an increasing appreciation that the immune system can be harnessed to control and eradicate cancer. And that has been shown now in several publications and also in the Nobel Prize that was awarded in 2018 to Jim Allison and Tasuku Honjo for checkpoint blockade. One of the major cell types that is focused on for cancer immunotherapy approaches is T cells. And so T cells are used in cancer immunotherapy because they are specific. So as the guided missiles of the immune system, T cells are able to target and destroy harmful cells in the body while ignoring normal healthy cells. T cells are also long-lasting. So after killing tumor cells, T cells can remain in the body as a memory population, poised to respond if cancer returns. And also T cells are very powerful in that a single T cell can kill 100,000 cancer cells. However, not all patients have sufficient tumor-specific endogenous T cells to re-engage with therapy. So for those patients, we've developed an approach where we restore the anti-cancer immune response with T-cell adoptive immunotherapy, which we like to call a living drug. So in this approach, we take T cells from the patient or a healthy donor. We genetically engineer those T cells to recognize the tumor by inserting a gene that is a cancer-targeting receptor, such as a CAR or a TCR. And we then infuse those T cells to the patient to find and destroy cancer. However, there are many challenges for T cells in the tumor microenvironment that can impede or prevent the T cell from destroying the tumor cell. Some of these obstacles include metabolic competition, such as reduced nutrients or oxygen, and so T cells aren't able to access those nutrients as the tumor cell usurps all of those available metabolic reagents. Also, T cells may have issues migrating to the site of the tumor, and tumors can upregulate inhibitory ligands, which bind to the inhibitory receptors on the T cells, causing the T cells to be shut down. So thinking back to our adoptive cell therapy approach, we have the opportunity to further engineer our T cells at the stage where we're engineering them to recognize the tumor, so we can help our T cells overcome some of these inhibitory barriers and be better equipped to find and destroy cancer. So the questions are, how can we best engineer our T cells to improve anti-tumor function in the context of a suppressive tumor microenvironment? But also, what approach improves function with minimal manipulation? So what is the best approach that we can do to afford the most benefit for these T cells? So as I mentioned, tumor cells can express proteins on the surface of the cell that shut down the T cell. So in this cartoon, we see the tumor cell has upregulated an inhibitory protein, shown in the light blue, that binds to an inhibitory receptor on the T cell, which is shown here in the dark blue. And binding of this inhibitory protein to the inhibitory receptor on the T cell results in the T cell being shut down. And with the T cell shut down, tumor cells can continue to survive and divide. So how can we engineer our T cells to overcome this inhibition? So an approach that we've worked on that has worked out really well has been to essentially replace a brake with an accelerator where we are rewiring inhibitory or death receptor signaling. So we take the inhibitory receptor ectodomain here in the blue, and we swap out the signaling components for that of a co-stimulatory receptor shown here in orange, which normally gives a boost to the T cells. So with that, we've generated a new fusion that has the inhibitory receptor ectodomain combined with the co-stimulatory intracellular signaling domain, making a new fusion protein we've termed an immunomodulatory fusion protein, or an IFP for short. So now when the IFP binds to the inhibitory protein on the tumor cell, instead of an inhibitory signal, T cells now get a stimulatory boost from the IFP, and they're able to once again kill the tumor cell. Some of the advantages of adding co-stimulation with IFPs are that we're abrogating an inhibitory signal and replacing it with co-stimulation in a cell-intrinsic manner. When we pick inhibitory ligands that are on tumor targets, we're lowering the threshold of activation to these tumor targets specifically. And also several studies now with CAR T cells have shown that the selection of different co-stimulatory domains can influence the phenotype. So depending on the co-stimulatory domain we pick, we can bias our T cells towards more of an effector memory or a central memory phenotype, and also influence the metabolism of T cells. So one of those barriers of the tumor microenvironment that inhibits T cells responding to tumor cells is FasL ligand. FasL ligand is normally expressed on T cells as a mechanism to kill cancer cells, but also to induce apoptosis in overstimulated T cells. However, tumor and other tumor microenvironment cells can also upregulate FasL ligand to induce apoptosis of tumor-infiltrating T cells. So the FasL ligand axis has been shown to be a limiting factor for T cell therapy. So there are several studies now showing that FasL ligand signaling to T cells can reduce their efficacy and essentially protect the tumor from T cell therapy. But FasL ligand is overexpressed in the majority of cancer tumor microenvironments. So shown here in this chart in the left panel, most of the tumors that were analyzed in this particular study have overexpressed FasL ligand expression relative to normal healthy tissues. So the way that we've decided to overcome the FasL ligand issue is to essentially cheat death, where we are replacing FasL signaling with 4-1BB. FAS and 4-1BB both signal as trimers. So by pairing the FAS ectodomain with the 4-1BB signaling domain, we're pairing two trimers, and we are essentially replacing a death with a pro-survival co-stimulatory signaling domain. FasL ligand positive cells in the tumor microenvironment induce lymphocyte death, but T cells can also upregulate FasL ligand after stimulation. So there's the potential here to generate a positive signal for T cells in an auto-co-stimulatory manner. So the first experiments that we did, we engineered T cells to express FAS4-1BB, and then we tested them in a proliferation assay. So in this assay, we load our T cells with a dye called CTV. And so if we leave those T cells unstimulated, they're very bright for the dye shown on the left panel. But as they're stimulated with a low level of tumor cells, so in this case, FBL, which is a mouse leukemia cell line, we see that our TCR-only T cells in the black remain undivided, they're not proliferating, but the TCR plus FAS4-1BB T cells are proliferating and diluting out the dye and shifting to the left side of that panel. So we were able to get proliferation where otherwise these T cells wouldn't be activated to a very low level of stimulation, so stimulating 100,000 T cells with 6,000 tumor cells. We also wanted to evaluate the ability of these T cells to kill cancer cells. So we used the IncuCyte instrument, which allows us to monitor T cells killing tumor cells over time with microscopy time-lapse images. And so the IncuCyte instrument sits within a cell incubator, and we're able to monitor how T cells kill tumor cells over multiple days. So to determine if IFPs could improve eradication of leukemia in culture, I developed an assay to really push our T cells, where we have our T cells in culture, and then we add in red labeled leukemia cells to those T cells. But then again, as I wanted to push these T cells, I add additional batches of tumor cells every 24 hours. So these T cells have to kill wave after wave of tumor cells. And we've dubbed this the serial killing over time assay, or the SCO assay. So these are some images from that assay. In the left panel is a picture taken from the beginning of the assay, where there are several red tumor cells interspersed with uncolored T cells. But by the end of the first 24 hours, most of those red tumor cells have been eradicated by the T cells. So when we compared our control T cells in the black with our T cells that had been engineered with the FAS4-1BB IFP in the red, with the first addition of tumor cells, we saw that both populations of T cells were able to potently kill off the tumor cells. However, when we added tumor cells a second and third time, we saw that the T cells that were the control T cells that did not express the IFP were starting to lose control of the tumor cells, and there was increasing tumor signal, as shown on the y-axis, and the increasing additions of tumor cells. However, when we engineered our T cells to express the FAS4-1BB IFP, we found that they were still able to potently kill off those tumor cells, and if anything, they seemed to be getting better at their job, as by the end of the 70-hour period, there are a few tumor cells left in the dish. We next wanted to examine if the FAS4-1BB T cells exhibited reduced FAS-mediated death. So by expressing the FAS1BB receptor on these T cells, are we providing a decoy mechanism to prevent FAS ligand to binding the endogenous FAS receptor? So to examine endogenous FAS signaling, we looked at caspase-8, which is part of the FAS death pathway. Following antigen stimulation, death signals are induced via the FAS pathway. So when we looked at active caspase-8 in our control T cells, shown here in the black, we see that there's upregulated caspase-8, indicating FAS pathway activation. And this is compared to FAS deficient T cells, shown in the gray. But when we looked at our FAS1BB T cells, we see reduced active caspase-8, similar to those in the FAS deficient T cells, suggesting that we are providing a decoy mechanism and these T cells are experiencing reduced FAS pathway signaling. So we found that FAS1BB improved the function of the T cells in vitro, but how are we altering T cell programming? As I mentioned at the beginning of this talk, we know that there are several metabolic barriers to anti-tumor immunity. So there's limited nutrients in the environment as they're being used by the tumor cells. And also T cells have been shown to have reduced number of mitochondria as they are exposed to the inhibitory tumor microenvironment. Several studies have shown that increased 4-1BB signaling can overcome some of these obstacles and improve metabolism. So 4-1BB stimulated T cells exhibit higher spare respiratory capacity, which is the reserve energy store for cells. And this has been linked to long-term persistence of memory cells. So in these two papers on the left, shown with 4-1BB CAR T cells, and on the right, shown with 4-1BB agonism via an antibody, we see that there's increased spare respiratory capacity for the T cells that have increased 4-1BB signaling. We saw similar results with our FAS4-1BB T cells, that they exhibit higher spare respiratory capacity, which was similar to those publications I just showed you. And also when we looked by microscopy at the number of mitochondria, we saw increased mitochondrial biogenesis in the T cells that expressed FAS1BB relative to our control T cells. So they are generating increasing numbers of mitochondria, and they have increased metabolism similar to previous reports. So we are getting a good 4-1BB signal through this IFP, resulting in remodeling of the metabolism of these T cells. So we're very excited about these results. We next wanted to test them in our in vivo mouse models. So we first tested the FAS1BB IFP in our mouse model of leukemia. So for this model, we inject 4 million tumor cells into the mice on day 0. By day 5, the tumor has disseminated, as is shown here in this IVUS image, where the tumor has spread out in the peritoneal cavity of these mice. So then we give them 1 million T cells after treating with Cytoxan, which is a chemotherapeutic drug, which we also use in our human patients. And then we assessed for survival over a 100-day period. So for this study, we did not give the mice IL-2 injections, which is a supportive cytokine for the T cells. So once again, we're pushing the T cells to do their job in the absence of support from IL-2. And we also gave a single T cell transfer. When we checked for persistence in the blood, we found that our FAS4-1BB T cells were more highly represented in the blood than our control T cells, supporting that they had enhanced persistence. And when we looked at the survival curve, we found that our TCR T cells that did not have the IFP, as shown here in the solid black line, did prolong survival, but ultimately did not rescue the majority of mice. So while we were getting a survival increase relative to mice that received chemotherapy only, as shown in the hashed black line, the dotted black line, we still did not rescue the majority of mice. However, when we treated with FAS1BB T cells, as shown here in the red, we did see a significant increase in survival. So by adding in this extra engineering of adding in this IFP, we were able to get significant increase in survival for mice that received these IFP positive T cells. So we were very excited about those results. The next hurdle that we wanted to overcome was to look if these IFPs could improve T cell immunotherapy of solid tumors. And the solid tumor that we focused on is pancreatic cancer. And pancreatic cancer is a cancer that desperately needs improved therapies. So in this chart, there are several cancers shown, And in the purple arrows, you see the changes in survival over a 40-year period. And for several of these cancers, there's been remarkable increases in survival with new therapies and new research as the treatment options are improved over time. However, pancreatic cancer is down at the very bottom, and there's very little improvement in the outcomes for patients with pancreatic cancer. Some of the challenges of pancreatic cancer are that early detection is uncommon. Pancreatic cancer tends to spread quickly, and recurrence is likely. So to model pancreatic cancer in mice, we use the KPC genetically engineered mouse model of pancreatic ductal adenocarcinoma, or PDA. And we use this model as it recapitulates the majority of human patient cancers in that it has mutations in KRAS and P53. And this is held under the control of a pancreas-specific CRE. So these mice spontaneously and continuously generate tumors from the epithelial cells in the pancreas. So with these mutations, they're constantly generating tumors. So we'll never be able to cure these mice, but we would hopefully be able to shift the curve with an effective therapy. This model has been successfully used to screen drugs to predict therapeutic efficacy in patients. So the results that they saw in this mouse model translated to humans. So another reason that we like to use this model. And also the KPC model recapitulates several barriers to T cell function of human PDA, including a suppressive microenvironment. So in previous work, mesothelin is a target antigen that has been shown to be reliably expressed in various stages of PDA, both in human disease and in the KPC mouse model. However, when TCRs were generated to mesothelin and those TCR T cells were used in therapy, we found that the tumor-specific T cells became rapidly dysfunctional in the tumor microenvironment. So these data are from day 8 post-transfer, and if we compare T cells pulled from the tumor versus T cells from the spleen, we already see a marked decrease in the ability of these T cells to produce effector cytokines. So the way that this model works is that we screen these mice by ultrasound to determine when tumors arise. So in a healthy mouse, we see, on the left side, we see a nice, normal, healthy, bright pancreas. But on the right side, we can now visualize tumors by ultrasound. And so these mice would be enrolled to study and randomly assigned to treatment groups. So for our initial studies in solid tumors, we enrolled mice to the study when the tumor was detected. We then gave them an enrollment dose of 10 to the 7th each of TCR T cells and also mesothelin pulsed splenocytes as an in vivo antigen boost for these T cells, again after Cytoxan, which is that chemotherapeutic drug. And then we took the mice down at day 28 and analyzed to see how our T cells were doing. And again, this is in the absence of IL-2 therapy. So when we looked in the tumor by histology, we saw an increased intratumoral concentration of IFP positive T cells at day 28. So we saw an increased number of T cells were able to infiltrate the tumor. And looking at it in a little bit of a different way, when we did a paired injection where we combined controlled T cells with our FasL and BB T cells at a 1-to-1 ratio and injected those into the mice, by day 28, there was a significant skewing of the FasL and BB T cells in the tumor. So although they started out a 50-50 mix, by day 28, it was more of an 80% of the T cells in the tumor were our FasL and BB T cells relative to 20% of the TCR-only T cells. So we next started the survival study. So again, we enroll those mice to study when tumor is detected by ultrasound. And then we give them an enrollment dose of 10 to the 7th T cells and mesothelin pulsed splenocytes post-Cytoxan. And then we repeat the T cell APC infusion without Cytoxan every 2 weeks for a total of 3 infusions, and then assess the mice for survival. So as we'd shown previously, immunotherapy with mesothelin targeted T cells improved survival from PDA relative to mice that received chemotherapy only, as shown in the dashed line. The solid black line showed the TCR controlled T cell therapy. But what was really exciting for us was to see the shift of the curve that we achieved when we treated with T cells engineered to express the FasL and BB T cells. So now we see a shift of the curve of a median survival of 37 days for the control T cells to 65 days for our FAS4-1BB T cells. And this was strongly statistically significant. We bled the mice for T cell persistence 28 days after their final T cell transfer, and we were unable to detect transferred T cells in the one surviving TCR-only control mouse. However, for all 4 of the surviving FAS4-1BB T cell recipients, we were able to detect transferred T cells. So we saw increased T cell persistence in the mouse and also increased trafficking, as we showed by histology and by flow cytometry. We've also now taken these studies into the ID8 ovarian cancer model, and we found similar results where we get enhanced survival of mice that receive our FAS4-1BB T cells in the ovarian cancer model. And we've also translated these experiments with human T cells, with human primary T cells. And we've, again, used the IncuCyte assay with PANC1 tumor cells. And so in the gray, without any T cells, we see that the tumor cells grow out over time. When you add in either TCR-only T cells or control T cells with just the mesothelin-specific TCR, or in the blue, a construct that just has the FAS ectodomain, so it's a decoy receptor, we see there is reduced tumor persistence, but really the big difference is with the red curve where you see that the FAS4-1BB T cells are able to eradicate the tumor cells in the dish. So we were very excited about these results, not only for the increased survival benefits that we've seen, but also in that we know there are several obstacles that inhibit the anti-tumor T cell response that I presented at the very beginning. We now know that IFPs have the potential to overcome several of these inhibitory barriers. So with our IFP T cells, we've seen enhanced metabolism, as well as reduced or ablated suppression by decoy. So we've seen reduced endogenous FAS signaling due to our FAS4-1BB expression. We've seen reduced exhaustion of our T cells. They're able to kill round after round of tumor cells, as we've shown in the SCO assay. There's a lower threshold of activation. These T cells are able to proliferate with a very low level of stimulation. And also, these T cells are able to get into the tumor and persist in vivo. So we have increased intratumoral concentration as well as persistence in our mouse models. So with that, I'd like to thank my lab. We just started on May 1st. And so Edison Chu is the first member of my lab. I'd also like to thank my former mentor, Phil Greenberg, and the group of researchers that worked with me during my time there, including Nicola Garcia, Cody Jenkins, and Katrina Polchikoff, and also our collaborators, Stan Riddell, Mike Jensen, and Ode Shapui, and our sources of funding, which include the Leukemia and Lymphoma Society, Juno Therapeutics, and then we have several new projects that we're working on now, and we're always interested in new collaborations and partnerships. So please feel free to reach out if you have some ideas or would like to work with us. Thank you for your time.